Yesterday, we saw how we can produce computationally realistic models of flowers from the, let's say, from the alpine point of view. But actually, as you also remarked, this needs to be based on some observation of plant growth. And it is assumed that before you get to the plant programming, you have a certain knowledge of how the plant grows and maybe to have quantitative measurements. So we are developing this type of models in the lab, and Fabrice Besnard is a biologist in the lab, in particular specialist of this small plant that is Arabidopsis thaliana. And Fabrice is currently proceeding to experiments in order to measure with kind of accuracy the development of the plant in order to be able to create these virtual models. So Fabrice is going to explain what he's doing about this. Fabrice.
Yeah, so thank you, Christophe. And indeed, so as you may know now, so I'm definitely more a biologist than an informatician. And so I know, I don't know the model very well, but I know the plant very well because I spend hours looking at it, how it grows and measuring it. And so I think we can have like a kind of fruitful collaboration to find ways to incorporate the knowledge of biologists like me into the model to make more realistic. And so, yeah, sorry, I have to move something. So here we have an objective, which is we would like our model to be realistic, and maybe just like as a very short introduction, we should say why we want such a model to be realistic. So very briefly. So first, it's not an objective per se, okay, because it depends on the model purpose. So Christophe very nicely Monday presented that you can make model for different goals. And so like the realism or the fact that your virtual plant will really looks like a real plant is not always a necessity, depending on what's the final use of your model. And so I was interested in this question. So for example, to explain like a flooring transition or inflorescence architecture, you could use models that are not like really realistic. And depending on how you and what you implemented in your model and what you're testing, this could be already informative despite the final rendering of the model is not really realistic. And in the case of ROMI, so this European project we are involved in with the guys from Paris, so DCSL. So we are using virtual plants for a precise objective, which is, and we have two sub-objectives where we identified that we can use virtual plants in a useful manner. So the first is a bit what Christophe also presented Monday, is to use virtual plant to make data augmentation and to create dataset, virtual dataset that we can use for training deep learning algorithm very efficiently and costlessly. And a second way to use a virtual plant is also to use these virtual plants to test, design, and prototype different tools just with one plant. So the idea is not to make like a population of plants like you would need for a proper data set with data augmentation, but just a few plants with typical architectural traits that we want to test. And then you can use these plants that are just available on one click on your computer to test your new algorithms to measure one trait, for example. Okay. So this is the both ways that we are following and why we are interested to use virtual plants in the framework of the ROMI project. And as I told you, then once you have an objective, you have to question yourself whether realism and the realistic rendering of the plant is useful or not for your objective. And here, I must confess that we don't really know this, and it's an open question. And I think it would be a very, I think, a good outcome of the roaming project if we can contribute to this question in the end, like how much realism impact this kind of objectives. But if we don't have the answer, we have kind of preliminary data suggesting that definitely more realism could help. So here I present you like on the, can you see my pointer, the mouse? Yeah, good. So here it's a zoom in a plant that was acquired with our robots, the plant imagers that we are using to reconstruct, to make 3D reconstruction of plants. So it's an Arabidopsis stem, okay? The branches, lateral branches have been cut. And here on the top, you see fruits, calyx. And in the program, so we use pipelines that are able to detect automatically, to segment the plant and to detect automatically the organs. Okay, so here basically it will be the branching point that will be discovered, and this branching point can be either branches that are cut here or calyx here. And once it is detected, so we use a representation where we highlight these branching points, so fruits or branches. And you can see that here you have an overlay, and this highlight overlays perfectly with the branches. So you can say, wow, it's really good. And for example, here I have another plant, which is zoomed out, when you can see that it's a full 3D reconstruction, and the point cloud is labeled differently if the part of the plant has been segmented as a fruit, pedicel, or stems. And which is very nice here is that like actually the point cloud, so the plant here is too thin to stay alone without to be tied by something. So here we have a wooden stick that holds the plant, and here a linker that also links the plant to the wooden stick here. And thanks to the machine learning algorithm we are using, so this wooden stick and this link are not recognized as a plant element by the deep learning network, and they are automatically filtered out from the analysis. So definitely, this shows that machine learning algorithms here are quite good to segment the plant and tear apart from an image elements that are part of a plant and not part of a plant. So that's a cool aspect of the story. But when you dig into details, sometimes this machine learning algorithm, are not working well. And for example, here you have another plant, which is not the plant that you see on the left-hand side. Another plant, you recognize the stem, you recognize branches that have been cut here, and at the just underneath the branch, you have a typical colline leaf, okay, that you see in every Arabidopsis plant. And here, when you proceed with the same algorithm, the leaf is recognized partly as a leaf at the beginning, okay, but the tip of the leaf is recognized as a fruit or a branch, or okay, and so which is an issue here. And for example, the stem here, you can see that the stem is highlighted here, which means that also the plant, so the algorithm identified that some pixels here should be labeled as fruit, whereas you can see with your human eyes that is not the case. Okay, so it shows you here that you have like some issues. So those machine learning algorithms are not perfect, and we need to improve them to increase our performances in terms of segmentation. And for example, if I make a zoom in our virtual plant, for example, oops, sorry. Can you see my screen? Yes?
Yes.
So I make a zoom in the virtual plant of Christophe, and you can see that definitely the leaves here are in the current model with which we trained this network. They are like quite simple structure with a flat, very simple shape that are not like really the shape of the curl leaf. So here there is an open question. If we make a new plant with modify modified here leaf shapes. Will this help this algorithm to make a better plant segmentation? And that's the same. So you can see that for the stem issue, maybe here the textures of the stem are not perfect. And if we improve a bit the texture or the shape and the dimension of the stem here, could we eliminate this aberrant segmentation of part of a stem as a fruit? Okay. No question on this part?
Questions? No?
So I move on. Don't hesitate to interrupt me because it's not as living as if I were in live with you. I have another data, but maybe David could comment. But with Ian, when we exchange it, so there are like the previous algorithm here that I presented you. It's a machine, it's a deep learning base, but the learning phase is done on 2D images. So each pixel on 2D images are recognized and labeled as fruit, pedicel, stem, etc. And David and Ian in the REM project are also developing another pipeline, phenotyping pipeline, where the deep learning process does not operate on 2D images, but operate directly on 3D point cloud. And the segmentation is done from the deep learning network directly in this point cloud. And from the first results they obtained, they told me that, for example, each time a stem is a bit bended at the end, like here or here, this causes some issues, for example. When the fruit angle is a bit weird too, and I think they had also issues on the diameters, for example. So, and again, as you can see here, for example, so like the bending of the different branches of the main stem is not really realistic. So it was like assumed to be like straight to the upper part, like but it's not. So like in the real life, stem and branches can be a bit more wavy, as you see. So maybe here the idea would be also to implement those kind of behaviors for stem and branches shape and curves to fit more realistic behaviors and hopefully erase some of the issues that we have observed during our segmentation tests. Okay. So then the question is, how was the model calibrated? So I think we should ask Christophe this because Christophe is the father of the model. But I think Christophe just calibrated it from previous data, previous work, and also his own knowledge of the plant because although he is not an experimentalist, he has been working for years now with Arabidopsis, so he has a good knowledge of the model. And if we look at especially for Arabidopsis, if there is like a previous work on trying to calibrate L system model of a plant, so we can refer to this article, which was published in 2005 now, which was in the group of Przemysław Prozinkiewicz that Christophe already presented. And so in this article, they definitely try to, so they generate a system model of Arabidopsis, okay, they describe it, and they use and they incorporate measures and countings that they precisely made on a real plants into their model. And so here, for example, so you have like each time a side-to-side view of on the left-hand side a real plant, on the right-hand side the model at hopefully the same stage. Okay, so here's a real plant, here's a model, here's a real plant, and here's a model. And the idea was to show that they succeeded in reproducing a realistic model behavior that really looks like a plant. For this, they incorporated measures on the main stem, so concerning the internodes, the leaf, on the lateral branch, also for internodes and leaves. They also make precise measurement of the flower's dimension and of the plastocron. But we can notice some limits to this work. So first, it was done in a particular Arabidopsis plant, which is a mutant background, because at this time, historically, many people are using this mutant background in biological labs. But it's so this mutant background is now a bit deprecated. So most of the lab now are not using it anymore because it has also some issues to use a mutant as a reference. So people try now not to use it so often, and so I think it would be good, like that we have a proper model that is really looking like the reference accession, the reference genetic genotype that most of the labs around the world are using. Also, they were cultivating their plant in very particular growth conditions, again, that are not like the standard growth conditions that are used by most of the lab. The calibration was well done, so and they explained it very well, but it was done only on 5 plants. So in their models, they only use a mean behavior, but they never incorporate the variance and the variability of it. And so I think that would be a very interesting work to question how we can integrate the variability of a measurement into a model. And also, so some of their measurements were done on pictures only. It's not clear how they perform it, and sometimes, so I think that they took a lot of pictures, and from the pictures they derived some of the measures they are relying on. And I think sometimes it's not the best way to do, I think. Okay, so now the general objective here of this calibration is to try to measure different parameters, to integrate them into an updated calibrated model of this reference accession. So accession is the term to name in Arabidopsis a plant which is a fixed genotype, and that should be the same in all different labs. And of course, once we have a updated model which is calibrated to be more real life looking, then we can test the effect on the phenotyping analysis by using like 2 types of trained model for deep learning algorithm, like one with a basic first uncalibrated virtual plant, the other with a calibrated virtual plant, and we can really test and demonstrate whether this calibration step has improved the phenotyping analysis on. And so today I will just present the data I collected, how we choose them. I will discuss how I'm trying now to exploit them. And of course we can discuss because, so this is a kind of the beginning of this work. And I think that maybe by discussing with you and getting your ideas, we can have like other ideas of other traits to measure precisely, on other way to measure it, for example. Is that clear? Yeah. So here I just present you an Arabidopsis plant, and I introduce a bit of different terms that I'm using and that I will be using again when I will explain you the different measures I made. So here, if we look at this inflorescence of Arabidopsis, I will present you the different part from the top to the bottom. So here, this is the inflorescence bud. So the shoot apical meristem is inside and produces the different flowers. And you can see that the youngest flower are inside this bud. And on the periphery, so you've got an older and older flower. And at some point, they are so old that they start to flourish. And so when they flourish, basically in Arabidopsis, it's when the petals go out of the sepals, which are the external, the most external organs in the flower. And so the petal grows so much that they push away the sepals, and it's correspond to a global maturation of the plant. So like for example, so here that the sexual that the time window where pollination can occur. So they enter our mature and they produce pollen grain, and uh, the the female genici is also ready to be pollinated. Okay, very soon after, like when you go down, and of course, when you go down the plant, you kind of go back to the past and see the evolution of the different flowers. So here you have a step where you have like a wilted flowers. It's where all the different parts of the plant, of the flowers, start to senesce, and and they separate. So you have an abscission between the organs, petals, sepals, and answers from the flower receptacle, which is the place where all those organs were attached before. Okay, so you all know that flowers can can wilt basically, but and it also happened like in planta here, and so you have a mixed here identity where the only part that does not fall is the female part that after pollination is transformed into a fruit. And so this fruit is growing out quite rapidly. And so here you have a phase where like the development of the pollinated seed inside the carpels here and inside the fruit are stimulating the development of the fruit. So the fruit is growing and growing and growing, okay, until kind of mature size here. And when it will stop growing, and in the end, so these fruits are dry fruits in Arabidopsis. So at the end of the life of the plant, they will just dry and open naturally, and which will be free the seeds that can be disseminated by this opening of the fruit. Oh, sorry. Okay. So of course you've got the main stem. So the different branches. So you've got different terms to name the branches in Arabidopsis, and so you have two type of branches. You have the branches that are like held by uh, by the main stem, and we call them the paraclades. Okay. And you can see that you have other branches that start from the base of the plant, this leafy part here, and which has what I call the rosette branches. Okay. So each paraclade here is subtended by a colline leaf. Okay. And so in the rosette, basically, you have all the leaves that have been produced during the first part of the plant, the first part of the life of the plant. Okay, and it's also where rosette branches emerges. Okay, is that clear for the architecture of Arabidopsis? It's not very complicated, I guess.
Yes. So colline leaf is all leaf which are not rosette leaf.
Exactly. So, and then you have different types of colline leaves. So, for example, you have the colline leaf, which are like at the branching point between the main stem, so the axis order 1 or 0, depending how you start to number. Okay, but you have also colline leaves like at the next branching order, like between a paraclade and the branch of the paraclade. Okay, so these, for example, are colline leaves, and they correspond to branching events that are like of a higher order. Is that clear? And so for this, we decided to measure different parameters, and especially like in the previous model, the model I presented you of the calibrated Arabidopsis plant, published 15 years ago. So most of the calibration was done like in the first part of the like during early time of plant development, and I think they stopped way before this stage, for example. So they never go beyond a certain stage where branches are emerging. So you can see like the branches here are quite small, and I think they stop at this stage. And my plant, for example, that I'm showing you is a bit older than this. Okay. So the idea of the model also and the calibration of the model would be also to kind of to be complementary to this one and to go at stages beyond and focus especially on the inflorescence, because in the rowing project we are not that much interested in the early parts like the rosette part of the plant, and much more like in the development of the inflorescence. Okay. And so we derived a lot of different measures that we could do on this different part of the inflorescence. Okay, and especially it mixes macroscopic measures and microscopic measures, because one of the questions here is, for example, in the inflorescence bud. So you've got many different flowers, and the idea was to have an estimate of how much flower do you have here, because like as you've seen, like all these flowers depend. So they are like a organogenesis event from the apical meristem. So like in LPI, you would say that at each step, okay, the apex generate an apex plus a flower. Okay, so a flower plus an apex and n plus one. Okay, and then this flower will grow. So at each step, you will grow. You will make this flower grow. And the question is how you you have to calibrate the rate where the growing rate of the flower to generate such a bud, which is realistic, that contains as many flowers as in real life. Okay, and this can impact the development of the entire stem, of course. And but just on pictures like this, you cannot do this. So for measuring this, what I did was to mix plants that I observe over time and plants that I dissect, so which is quite destructive. But for some plants, so I measure the length to have an idea. I measure the length, I count the different flowers that have been produced. So I know a bit of the history of this apex. And then I dissect the plant and I count all the flowers that belong to this inflorescence bud here to have an idea of how much they contain. And then for, it's a bit the same, like here, we don't know exactly, and in the virtual model, it's not really realistic, like how long it takes for a flower to go out from this bud, to flourish, to wilt, and then for the fruit to grow until its mature shape, like how how big of the plant it will take, like these different parts here. And so the idea was on every stem to count it, like, and is it stable? Does it change over time? So do you have always the same number of open flower, or do you have like a difference between like young stems and old stems? Okay. Okay, and so the idea was, so I don't know if I describe all these different things, but the idea was to derive basically a mixture of counting and growth rates. Okay, and the idea was to measure directly with a ruler, for example, the different lengths at different time point. So to make a time course experiment, of course, and also some complex phenotypes, like I was saying, I was interested in the bending and the curve of the different branches here. It's not easy to measure, but if you take pictures, then you can try to use the picture to calibrate this. Okay, so I made a mixture of measuring and just noting all my counting and measure in files, and also taking a lot of pictures that we can use our raw material to calibrate complex phenotypes such as shape, curves, etc., etc. And finally, as I told you that I was interested in getting the variability of the plant. So the idea was not to take only 5 plants, but try to take a bit more plants that was done before, and so that we can use both the mean behavior, but also the variance for each of those traits. Okay, so here it's so what I did so far was like 2 experiments. So here you see it's a way of representing it. I will explain you how it goes. So here it's a batch. So each line is a plant, okay, and each column are a day of observation, okay. And you see that. So I have 2 batches of experiments, okay, And for those 2 batches of experiment, plants were synchronized or hopefully synchronized. And I observe them at different days, okay? And sometimes, for example, this one, I stop them because I dissect them to explore their inflorescence bed, for example, okay? So this plant is stopped till the end. So in the end, you have only a few plants, like for example, only 5 plants over 19 here that I could observe till the end where I have an entire time course. And for the other, I have so in the earliest day, I have more plants for every time point. And as long as the experiment goes, I have less and less plants, of course. Okay. So I did it twice. In the 2 batches, here I focused on different traits of the plant. For example, here I focused more on the rosette and here more on the different inflorescences. Okay. And so I decided, because all this experiment, it takes some time. It can be very time consuming. So I decided to not to measure every day, but to measure every 2 to 3 days, to be quite flexible. So it was an assumption that anyway, so the plant is not growing so fast that even with this regularity of measure, I could have like good data, good enough, let's say, to infer the parameters I wanted, like growth rates. And so, but it's an open question, so I don't know. So we will see from the results if it's working or not. Okay, so here I give you some examples of the picture I took and also of the plants. So for example, in the so from the same batch, so in this first batch, it was quite interesting because I don't know what happened. So something happened. It was not usual, but I have a lot of variability during. So I think my seed stock was a bit old. That's the reason, and I had quite some variability in the flowering time here. So for example, you see that at this day, so this plant was just bolting. So bolting phase is the phase where the stem, the main stem, is growing out of the rosette. So just before you have only a rosette and the plant is totally flat, just at the ground level. And the bolting, it's where you have the floral transition that just happened before, flowers that have been produced, and the first internodes below the first paraclades starts to elongate, and then you see the stem emerging. And you see that this plant is just bolting here, while this plant has already bolted, and I think for like maybe 1 week or like, so you had a significant desynchronization of this batch of plant. But at the same time, I think it was good because you can see whether you can compare the growth rate of those different plants in the end. Okay. And then, so for example, so I told you I was interested first in the inflorescence bud. So, and if I come back here, so I dissected quite a lot of different plants. So from this, you can make statistics and about how much flower buds there is like in an inflorescence bud. Okay, so if I so here you've got a box plot, which is a descriptive statistics for all the plants I dissected, all the different stems, and you can see that in one single inflorescence bud like this, you have like between 30 and 35 young flowers that are present. Okay, with internodes that barely not elongate. That's why they are clustered together in this inflorescence bud. Okay, and
You count by naked eye or do you count it under a microscope?
no, under a microscope, yes. So you dissect. So the first one, you can dissect them using your naked eye. But as soon as they get like too small, then you go under a binocular. So it's not a microscope; it's just a binocular, and you count them. And also here, there's a question of definitions. So I stop the dissection until a certain stage of flora development. So when I say there is like 30 to 35 flower, so for example, if I make a here, I start counting at the first flower after the last or the first, depending how you count, but open flower. Okay, So I remove the open flower and then I start counting because those, even as they are a bit detached here, so generally like 1 to 2 flowers that are a bit bending out of the bud. But if you look at their internode, it's not elongated. So it's just the bending of the peduncle, basically. So I start counting at them until a stage which is well defined by developmentalists, which is the stage 4 of the flower development. Okay. And beyond this stage 4, you can see also some flowers that are at even younger stage, but they are very close to the meristem. So then you have to define where you consider that you belong to the meristem and when you're out of the meristem. Okay. Then, for example, here you have a so most of the plants I dissected were coming from the main the main paraclade, so the main stem, but I also dissected from the different paraclades, and in the different paraclades it's a bit the same. So they have like between 30 and 35 flower buds. Okay, and interestingly, so and this is quite stable over time. So I made different statistics because you can plot the, for example, in the x-axis, so you can plot the total length of the stem. Okay, and there is not that much correlation. But a good way to see a correlation is so here you count the age of the plant. A good way to count the age of the plant is to count how much fruit it has produced here. Okay, so and to count how much fruit it has produced. So if I go back to here, for example, so this fruit is the first fruit of this stem. Okay, so here, for example, I can count that just before the inflorescence bud, you've got 1, 2, 3, 4. See 4 here, 5, 6, 7, 8, 9, 10, maybe 11, 12, 13, 14 behind, and 15. So I would say that roughly like this, looking at this picture, this last open flower is the 15, the number 15. Okay. And of course, if you come like 2 weeks later, so you have a very long stem with a lot of fruits, and maybe the last open flower will not be the number 15, but the number 45. Okay. So here, that's what I plot here. And you can see that at the end of the life of the plant, when you have like more than 50 flowers that have been produced and transformed into fruits, the number of flowers that you have in the bud is starting to decrease here. So we can make so basically the bud is trying to get empty, or you have less and less flower, and we can see at the end, very end of the life of the plant, it's very often that you can see that the bud is only is completely exhausted, and you see only 2 or 3 flowers. So the bud is consisted of only 2 or 3 flowers. So I think I started to catch this moment when after a stationary phase here, basically what I imagine here is that the rate of flower that goes out from the inflorescence bud is higher than the rate of production of flower inside the bud. Then it gets depleted. And at the opposite side here, so here, for example, just when the plant, so you have no flowers that have been produced here, or no flowers that are opened, and I stopped some plants at the very early stage, so just before bolting or at bolting, and I dissected the number of, I dissected the meristem to get the number of buds here, and you can see like a building phase where you progressively reach 30 or 35, and I think that it shows you that before you see 1 or 2, or before you see the first flower open, it means that you have to wait for the meristem to have produced around 30 flowers at least, and sometimes more. Sometimes more. Okay. So for example, that's the kind of investigation we can make here. more basic counting. So it's quite easy to count the number of colline branches. The number of rosette leaves, it's more complicated. I will show you pictures how you can count this. And the number of rosette branches here. And here I showed you that I have two different batches of plants. So which are two plants that I, different experiment, independent, that I made at two different times. And so you can see some variability. So definitely this batch is more variable than before. I don't know why. So you can see an experiment effect. And for each batch, you can see that yes, the number of colline branches is between let's say 3 to 5. Okay, the number of rosette branches is between 5 and 6, let's say. And but the number of rosette branches increase with time, so it's much more difficult to count than colline branches, because the number of colline branches for the main stem it's fixed, and once it has been produced, it won't change over the time, the lifespan of the plant. But for rosette branch, so they emerge, new branch emerges from the rosette with as the plant gets older. Okay. And for example here, so what I did was to so I plot the growth curve of the different branches of the plant. So for the two plants I presented you before, so in red that's the main stem. P1 to P5 are the different paraclates, okay, and R1 to R5 are the different rosette branch that emerges from the rosette. So as I told you, so if you stop the experiment at 45 or let's say 48 days, you will not see those branches. That's the same here. So if you stop before, like you will not see those branches because they emerge later. Okay. But it's interesting to see that, like at least from an airplane here, the growth curve are very similar between different branches. So the growth regime. So what I conclude from this is that the growth regime between the main stem and the other branches are quite equivalent. Okay. So another trait that we can try to calibrate is the flower production rate. So here, what I did was so every day I count the total number of here of the calyx or fruit that are on the stem. And of course, so this so it's cumulative. So you expect that this number will grow, and if you plot it as a function of absolute time, so you see my two different experiments, okay, with globally experiment 1, which was delayed compared to experiment 2. Okay, but the curve seems to be the same. And the interesting point here is that, so this number of flowers, I counted them, or fruits, sorry, I counted them on the main stem. Okay, and if you, instead of the absolute, so day DAG is day after germination, so that's the start of the experiment. And if instead of this time, absolute time, if you plot the length of this main stem, you can see that then there is no longer differences between the two experimental batch. So the fact that this one is delayed, it's not an issue. Basically, this rate of its flower production rate is the same as the experiment 2, and basically there is a clear correlation between the flower production rate and the growth rate of the stem. And you can see also this kind of offset. So before a certain size, there is no hope to see any fruits like so. Yeah. Okay. Another question I had was whether you had a constant number of open flowers, wilted flowers, and developing fruits here. So I have statistics on this too that I can comment briefly, but basically it's the same. So you have a lag period, and then when they start, it's a long stationary period. And even if you have like a old or young, so you don't see this correlation as we see before. Even if you have young, meaning short stem, or old, meaning long stems, you always have between at the same time 4 to 6 open flowers here and 2 to 4 wilting flowers. Okay. Okay. So right now I'm still analyzing this. Yes, and basically that's the kind of data I would like to have is the axis growth rates for all the plants I measured. Also to infer the plastochrone. So from this, infer the plastochrone and the plastochrone in the main stem and in the lateral branches. And one thing I would like to do also is to determine precisely how much delay there is between the flowering of the main stem and the lateral branches, and on the main stem and the paraclade and the rosette branches. And I also make some measure of the diameters with a precise device to measure distances. And I measured and collected different diameters, like of the main stem at the base, at the top, just below the inflorescence bud, to have an idea of the the conic decrease of the main stem. I measured diameters of floral peduncles and fruit peduncles of the different fruits too, etc., etc. So the idea is to calibrate a bit the different cylinders that we are using for modeling this plant. Okay, and just maybe to finish, I will present you. So all this I measured using or counting or like, and I wrote down the results of my measures. And I also have a lot of pictures now that we can analyze. So I will show you, for example. So here, to count the number of rosette leaves. So what you do is dissecting leaves, dissecting the rosette into pieces. So you separate every different leaves, okay, so that you can really see them and count them precisely. Because when they are all packed together in the rosette, it's very hard to get. And here, for example, the rosette is it's a messy part, but here maybe we can have a better construction of the rosette if we have a clear idea of its a different constituent and its different parts. And especially you see that you have a which is already well documented for Arabidopsis, but an evolution of the flower of the leaf shape depending on their age and their order of formation. So here's are the two first leaves that were produced, and then in this order, you can see, for example, that you have a long petiole at the beginning, and then the petiole gets almost nothing. Inside the rosette, so these are the leaves that have been produced by this shoot apical meristem, the main stem here. But rapidly, at each at the junction between this leaf, for example, and the stem, you have a new meristem, and all these leaves are secondary leaves that have been produced by these different meristems that were at the junction between leaves and stem here. Okay. Okay. And for example, if you zoom in here, you will see that this here, for example, you have a rosette branch which is ready to grow out, that you cannot see in the rosette, but it's really preformed here, and it will grow out later. And those are the paraclades. And here you have a nice view of the shape of the colline leaves here. Okay. So you can do it for a different plant at different ages. For example, this one was a bit younger than the previous one. Okay. And you can do it, as I told you, as different ages of the plants. So you can compare the different sizes of the different parts of the plant. Like here, for example, you start to see the rosette branches. So you can see the very short rosette branches. This one has emerged, okay, etc., etc. Here you can compare the differences between the main stem, the paraclade, and the rosette branches. what I did also was to focus on cilic development. So here I took an entire stem from the inflorescence bud, and I just align the different fruits so you can see the evolution of flower to fruit development and all the phase of fruit maturation. So you can use this kind of, so I have several of it. And so you can use this for several purposes to calibrate the growth rate of the fruit for the flower to fruit evolution. And also, like, you can see that the fruits, they can have like variable shapes, and you can try to capture this variable shape in fruits that hopefully will make the segmentation pipeline more robust to variation in fruit shape. Okay, and since I told you I was really interested in leaf shape, especially conine leaves. So here, for example, I just took some picture of conine leaf without, so just separated, but like is in their native shape. And here I just spread them on a tape here, so that you can have an idea of their fully flat shape and how this flat shape curl into this final native shape. Okay, so here it was just an overview of the work I done to, or I initiated to calibrate. So now I have different data. It's still ongoing, but I will have more and more. And now the question is how I integrate this data into the model. And then we can discuss whether it's just a question of giving the right parameter into the model, or maybe if for some point we need to modify the way the model is implemented, for example. And of course, so from this, we can also discuss whether I should collect other data, because my first two experiments do not provide those data. Okay. And as a perspective, I think because the RAMI project is to use virtual plant, okay, to ease the process of phenotyping, okay, and to avoid doing time-consuming measurements. And here, what I'm doing is that, okay, I'm doing time-consuming measurements to make a good model. So it's kind of a nasty circle here. And so I think in the future, we could imagine that one good tool for a virtual plant would be to use robots and phenotyping tools that we are developing, such as the one we are developing in RAMI, to help modelers like extracting interesting parameters. So some will be inaccessible, like this dicycling and inflorescence bud, but some other could be quite easy to access from pictures, like 2D pictures, like we are doing. And we could imagine to make a kind of tool suits that would ease the task of modelers to extract important parameters that will make their model more realistic. Okay, and then of course you make a virtual circle. You make a positive virtuous circle between. Then once you have a better model, you can use your model to make even better extraction and better phenotyping experiment. Okay. So I think that's what I wanted to present you today.
Thank you very much, Fabrice. Do we have questions? Different ways to measure plants.
Maybe it will seem you a silly question, but how could you control if the Arabidopsis plant will have one branch or three, as we can see?
You don't control it, basically. It's
okay.
So,
do you just get if there are three branches and you get the two other branches?
I don't get your question. So
I'm not biologist, okay? So sometimes I see the Arabidopsis with one branch and sometimes with 3 branches.
Yeah.
How can you get only an Arabidopsis with 1 branch?
Okay. So in our, so definitely the number of branches depends on a lot of factors, genetic and environmental. In our culture condition, and normally like in even in the world, if you look at our Arabidopsis growing out right now, because that's a good season outside, you will see that they always have branches, paraclad branches and rosette branches. So in their natural habit, generally they make branches. And so in our setup in Rami, most of the time we just cut the branches so that we have a simplified architecture. And so we so we make this. So to have like the typical fishbone, as we say, a fishbone is just the main stem, and the bones of the fish are the different silics. Okay, so it's a very simplified architecture. So we operate on the plant to cut the branches and simplify the architecture. But in the native development, they will make branches, and the number of branches, for example, in the lab, we can play with it. So different genotypes, different accessions of the plant with different genotypes, so we have more or less branches in the same culture condition. And also in the lab, we can play a bit with the culture condition to have more or less branches. Even if we, for one given genotype, and in apparently identical culture conditions, so there are always micro variations, but when you put your plant in a culture chamber, okay, but you try to have homogeneous culture condition. But even this, so you try to control the genotype, so the plants are the same in terms of genes, and the environment, so you put them in a culture chamber when you try to have a homogeneous culture condition, you have variation. So some plants will have 3 branches, and some of the plants will have like 5 branches, for example.
Okay.
And this variability, at least we cannot explain it. It seems to be stochastic, or a bit of stochasticity. And I think so it will be good that in our model, so we can capture this variability. At least all the 3 sources of variability, it would be good to capture them in the model. So we could capture the what we call the stochastic variability. So if you fix the genotype and the environment, you still have some variation. Then we could capture the environmental variation. You model an environmental variation when you know that it has an impact on the number of branches, and you can reproduce it. And we could also model the genetic variation. So you model 2 genotypes, ones that make few branches and others that make a lot of branches, and you can recapitulate it. So then it means that at least you have understand the underlying processes, I guess.
Okay.
Other questions? Maybe Fabrice, a general question on what is going to follow up from this work. Are you far from having the full pipeline ready for measuring plants? Is it like in the next weeks, or is it in the next year or years?
Do you mean by pipeline?
The pipeline, which is the robot looking all around the plant, taking pictures, and then sending the information to some software interpretation algorithms, and then getting out the different branching angle and the segmentation of different organs.
So now the pipeline is ready, definitely. So it's working. And that's what I presented first. So sometimes it works well, and on some plants, it does not work well. And so we are at this step where we have, let's say, a success rate, which is not totally satisfying. And so the idea is to improve our algorithm to have like a better success in the phenotyping task. But I would say, and thanks to the work of Nabil and Joe, especially, like all the different parts of the pipeline from the robot up to the final extraction of angles and internodes. So is working well. Like we can pipe the different parts and get a result. The question is, are we satisfied by the result? So sometimes yes, on certain plant it will work, and sometimes not. So that's why I'm interested in having a. So once we have identified the issues, like here, for example, leaves are segmented as fruits. Okay. The question is whether more realistic models could help having a better training phase. And if we have a better training phase, maybe the same, exactly the same pipeline, but with another training model, will be able to have a better success in terms of segmentation.
And with respect to your virtuous circle in the end, is it the case that if you, for instance, you manage to get very successful extraction of geometric parameters with one accession, then do you expect, or is it part of your expectation, that if you go to other accessions, it won't change the main behavior of the core algorithms, and then you may be able to extract the main geometric features of another accession or other accessions?
Yeah, definitely. That's something we would like to test with right now, especially with Marie, who is more like now she's dedicated to use a robot and to produce data, to make acquisition and to use the robot in different conditions. And so one of the thing we would like to test would be to take different accessions with different morphology, okay, and see whether the pipeline are robust to this change in morphology. But also we could go quite far in the change in morphology, because you could take other plants. And so of course, if we take completely different plants, there's not a great hope that it will work. But we could take like other brassicaceae, so other species, but quite close, that have exactly the same type of inflorescences with you know, a raceme, calyx. But you know that in brassicaceae you have a big variety in the fruit shape. So you have elongated or heart shape. Okay, and so you have very common brassicaceae growing out like in our surroundings. So it would be so one of the tasks we are right now trying is to take some of these brassicaceae and make acquisition and see how good is the phenotyping task with our model, which have been trained on Arabidopsis only.
Okay, thank you. Any other points or questions? Okay, so thank you again, Fabrice.